Hi, welcome to our channel of IGNOU Audiobooks, Indira Gandhi National Open University, School of Sciences, SOS, Master of Science, Zoology, MSCZOO. Semester 2, MZOE002 Animal Behavior and Animal Welfare Ethics. Block-1 Introduction to Animal Behavior, Concept and Evolution. Unit-3 Complex Behavior and Patterns, 3.1 Introduction. In nature we observe beautiful interactions among various animals and the environment in which they live. Baby birds as soon they are born start begging for food. Newborn puppies even with their eyes closed are able to suck mother teats. A praying mantis sacrifices its life for mating and weaver bird toils for days to build a beautiful nest for female. This often makes you wonder what mechanism is driving animal to perform such complex task. How they know what's a suitable response for a given environmental cue? How all members of a particular species are able to behave alike under similar conditions? In this unit we will try to answer these queries by understanding inbuilt behaviors of organisms. All animals rely on their instincts to react to environmental signals. It is the instinct that keeps animals alive and drive basic impulses such as foraging for food, hunting, finding mate and escaping dangerous situations. Instinctive behaviors are inheritable and expressed since birth or the first time an animal encounters a stimulus. They are different from acquired behavior that an animal gains during its lifetime and is based on experiences. We will study the complex neuronal mechanism controlling innate and instinctive behaviors. We will learn about the stereotypical behaviors like reflex and motivation and understand mechanism of fixed action patterns. We will also analyze manipulations of key signals by opportunistic species via code breaking and brood parasitism. Last but not the least, we'll study the beautiful mechanism of mimicry and its various types in nature. In essence, this unit will explore the complex behavior and patterns among animals and evolutionary adaptations pertaining to them. Objectives. After studying this unit, you should be able to define innate and instinctive behavior of organisms. Describe various subtypes of innate and instinctive behavior in animals. List the theories related to innate and instinctive behavior. Explain the innate releasing mechanisms. Elucidate open and close IRM. Describe key stimulus and stimulus filtering. Identify and explain supernormal stimuli. Explicate characteristics of fixed action patterns in animals. Determine the evolutionary features of fixed action pattern. Describe mimicry and its subtypes found in nature, and explain mimetic releases and their significance. 3.2 Instincts and innate behavior. Innate behavior is hardwired stereotypical behavior exhibited by animals. And instincts are those behaviors that animals display when they encounter a stimulus for the first time. Many times they are being used interchangeably, but there is minor distinction between them. Instinct has a huge innate element in it, but it also improves over the period of time with multiple exposures. In a nutshell, these two behaviors shape the complex behavior and patterns of animals. 3.3 Innate behavior. Innate behavior is based on the genetic makeup of an organism. It is hardwired in the nervous system and is heritable in nature from parents to offspring. A behavior is called innate behavior when animal repeats the behavior in same manner again and again with every exposure to the stimulus. Stimulus in this case triggers an unstoppable chain of responses that leads to final behavior. This behavior is also termed as stereotyped or inherent or inborn behavior. Example, the sucking reflex in newborn mammalian babies, grasp reflex in babies, Fig. 3.1, 3.3.1 Characteristics of innate behavior. This behavior is achieved by complex neurophysiological mechanism. This is predictable in nature. They are species-specific response. They are also sex-specific response. They do not have any effect of experience on it. Response will be same when exposed to stimulus for the first time or the 100th time. 3.4 Types of innate behavior. 3.4.1 Spatial orientation. This involves placing an organism in definite space in relation to a stimulus. It includes kinesis and taxis. By this mechanism, an animal orients itself in response to either single or sometimes multiple stimuli present in the Environment. Example, movement of flatworm Dendrocoelum lacchum away from light. 3.4.2 Reflex. Reflex is simplest and fastest response against a stimulus. It is characterized as a spontaneous, swift sequence of action that occurs in response to a stimulus. A reflex is directly proportional to the strength of stimulus. Example, knee-jerk reflex, Fig. 3.2b, when tapped with reflex hammer just below patellar cap, below kneecap, leg spontaneously produces a kicking action. Reflex arc and its components. Reflexes are outcome of neural mechanism and its neuronal path is termed as reflex arc. It has 5 components, Fig. 3.2a. Fig. 3.2a, reflex arc and its components. Look at the screen. Sensory receptor, the external or internal receptor cells that detect a signal. Afferent, sensory, neuron, this carries signal from sensory cells towards central nervous system. Interneuron, it connects sensory and motor neuron. Efferent, motor, neuron, it carries response impulse to effector cell. Effector organ, the muscle or gland that responds for the given signal. Fig. 3.2b. Reflex arc of patellar tendon, knee jerk reflex. The knee jerk reflex, Fig. 3.2b, is special as instead of connecting it with interneuron, it directly connects with efferent neuron in spinal cord forming monosynaptic arc. Any reflex mediated by multiple interneurons is called polysynaptic arc. 3.4.3 Motivation. Fluctuation in physiological state or need of an organism leads to drastic variation in response to the same stimulus. Among those varied responses, the response that is directed toward a specific goal is called motivation. Example, a herd of lions when hungry attacks buffalo, Fig. 3.3, wildebeest, or other prey, but same lions when not hungry pass by them without even noticing them. In this case, hunger causes food motivation. The stimulus which is sighting of buffalo is same in both the cases, but the physiological state of lions, i.e. empty a full stomach, will change lions' behavior towards the prey. Theories of motivation. Lorentz hydraulic model of motivation, according to Konrad Lorenz, for each action driven by innate behavior in organism, there is a specific reservoir in brain with action-specific energy buildup. This reservoir is closed with valves that can be released by a suitable stimulus. The external stimulus, such as presence of food, predator, mate, or internal stimulus such as glucose level, hormone, etc., works as causative factors or weights on scale pan pulling valves of the reservoir. This helps in releasing the build-up energy from the reservoir. Fig. 3.4. Look at the screen. Fig. 3.4. Lorenz's hydraulic model of motivation. For example, if an animal did not have food since long time, the energy build up is high in the brain, hunger, and sighting of prey. Food works as weights that pull and open up the valves and release the build-up energy. This leads to action patterns involving chasing the prey or grabbing food till the animal is fed and full. Tinbergen's hierarchical model of motivation. Nico Tinbergen improved upon the Lorenz's model of motivation and added complexity to it. According to Tinbergen, motivational impulses accumulate in the brain and nervous centers and they are held in check by multiple blocks. The nervous centers with motivational impulse are arranged in hierarchical manner containing its own block. The innate releasing mechanism removes these blocks and permits the flow of energy to next center till all blocks are released and behavior is expressed. This model showcases that behavior is a result of complex neuronal transmission and can be grouped along related behaviors. Fig. 3.5. Look at the screen. Fig. 3.5. Tinbergen's hierarchical model of motivation. 3.5 Instinct. Any behavior performed by an animal on its first encounter with a stimulus, without any need of learning, is termed as instinct. These behaviors are similar in all members of the species, but still has room of improvement on repeated exposure to stimuli. In this way, although it is controlled mainly by the innate elements but still has scope of improvement based on experience of the organism. 3.5.1 Examples. Nest building behavior in tailor birds. Tailor bird's nest, Fig. 3.6, is a Complex structure that requires sewing two large leaves together by punching holes with beak at the margins and threading them together using strands of cocoons, spider web or pieces of artificial strings. This structure is made into a pouch by pulling leaves tightly while sewing. The bottom of nest is cushioned using twigs and grass. The young male tailorbird, in a bid to find a mate for the first time, needs to build a nest to attract female. The first timer has never seen an older bird building a nest and still manages to sew two leaves together to make a nest. It might not be as perfect as the older birds, but still comfortable enough to take care of the eggs and raise chicks. Fig. 3.6. Construction of orb-shaped spider web. The orb-shaped web, Fig. 3.7. Constructed by spiders all over the world, is the product of stereotypic behavior patterns and very important for catching prey and survival of the spider. Spiders have abdominal silk glands which secrete silk threads that are spun into a web by 3 pairs of spinnerets. The initial step of making orb web involves first making Y-shaped scaffold and then adding frame and radial spokes to it. Fig. 3.7. Later auxiliary and stick spirals are added to it. This complex technique is put into action based on spiders' instincts. Look at the screen. Fig. 3.7. European garden spider constructing orb web. 3.5.2 Instincts, theories. Many scientists tried to understand and describe instinctive behavior. Initially, instinct and reflex were clubbed together, even used interchangeably. Theory by Jean-Henri Fabre. Jean-Henri Fabre was French naturalist who studied small animals including insects, birds, and compared their behavior with that of human. According to him, major difference is that humans are capable of making conscious decisions and behave accordingly. Animals, on the other hand, show unconscious behavior traits in response to their environment and repeat it every time when exposed to same stimulus. He observed complex behavior like taxes, molting, mimicry, maternal behavior, and playing dead as unchangeable behavior in animals. He observed various species of wasps catching its prey and identified species-specific fixed patterns. With the exception of one species, most wasps do not rearrange this pattern in response to modified signal. Based on his observation, he proposed instincts are fixed patterns that are shown by animal in response to a signal that do not change even after environmental situation has been altered. Fabre was anti-evolution theorist. He believed that instincts are fixed and can't evolve with time. Theory by Wilhelm Wundt, regarded as father of experimental psychology. He studied both animal and human behaviors. He considered instinct as unconscious process that is product of sensations and emotions. He correlated baby's facial expression when introduced to new sensations like sweet, sour or bitter taste. Each sensation is able to elicit a particular expression, and every time same expression is seen with the same sensation. He used instinct and reflex as interchangeably and inducted many reflexes under instinct only. Wundt explained instinct as survival mechanism designed to protect babies from eating something unpleasant, possibly toxic. He explained that instinctive behavior gets naturally selected in a species due to survival benefits it provides to the species. Theory by William McDougall. He associated instinct with emotions. He explained it as a behavioral pattern seen in animals and not applicable to humans. Theory by Abraham Maslow. Maslow proposed instinct behavior so fixed that it cannot be overridden under any situation, since humans are capable of overriding their innate drives. In many situations, instinctive behaviors are not applicable to humans. Theory by Konrad Lorenz. He, along with Nicholas Tinbergen, established the distinction between instincts and learned behavior. He studied imprinting in goose hatchling and gave the concept of sensitive period. In this behavior, newly born chicks of goose imprint on the first moving object they see post hatching as their mother and follow it everywhere. Imprinting itself is a learned behavior, i.e., chick learns to recognize its mother, but what is considered as mother, first moving thing, is instinctive in nature. Theory by Nicholas Tinbergen. During 1950, Tinbergen, along with A.C. Pudek, studied food begging behavior in herring gull chicks. Fig. 3.8. It was observed that, as soon as gull chicks were born, they would start pecking the red mark present on the tip of its parent's beak. This initiated regurgitation of half-digested food from parent's mouth into the chick's mouth. Fig. 3.8. All chicks show this behavior as soon as they are born to get their meal. To prove that this behavior is an instinct, Tinbergen conducted experiment using cardboard model of birds. Look at the screen. Fig. 3.8. Adult herring gull feeding its chicks regurgitated food. Experiments. Tinbergen and Perdeg designed various bird beak models made up of cardboard which were grouped into 3 sets. Set A. Shapes of beak. Different shaped beak models were presented in front of the chicks. Fig. 3.9. The normal beak shape was used as control. Rest of the shapes varied between broad to elongated one. Fig. 3.9. Model for shape of the beak. Note, the line indicates the amount of response. Result, chicks pecked the elongated beaks far more than the broader one. In case of the thinnest model, the response from chicks was almost double as compared to the control one. Set B. Color of patch on the beak. In this experiment, the beak shape was kept same, but different color of patch was used on the yellow-colored beak. Fig. 3.10. Look at the screen. Fig. 3.10. Model for color of patch on the beak. Note, the line indicates the amount of response. Result, although the chicks pecked the red patch the most, difference was not much as compared to other colors like blue or black. Set C. Contrast between beak patch and beak color. The beak shape was kept same, but different color for patch was used on the gray-colored beak. Fig. 3.11. Fig. 3.11. Model for contrast between beak and patch. Note, the line indicates the amount of response. Result, chicks pecked the patch, which had the best contrast with the beak color. Inference. From the above experiment, it was deduced that chick behavior is highly influenced by the parameters of the model. Long elongated beaks were preferred over short broad one. The red color patch on beak gives best response and better the contrast between the patch with respect to its background, more will be the response. All of these parameters were already fixed in the nervous system of chick and it responded to them even at the first encounter. The subnormal stimulus will get lowered response, low contrast, whereas supernormal stimulus, highly elongated beak, will receive exaggerated response. 3.6 Key stimulus. Stimulus, any kind of signal that can be detected or sensed by a particular animal is called stimulus. This includes light, odors, chemicals in environment, sound, vibrations, touch, heat, electric energy, etc. Key stimulus, also known as sign stimulus, it is any stimulus that will initiate fixed action pattern, FAP, in an organism. If key stimulus is from one animal to another, it is called releaser. Examples, in case of herring gull, the red patch present on the beak works as sign stimulus that initiates fixed action pattern of pecking in the chicks. 3.7 Innate releasing mechanism, IRM. When sign stimulus is detected by the receptor present on the animal body, it will initiate a neuronal network that will produce fixed action pattern in response to key stimulus. This whole network of complex signal transmission from environment to brain to animal body resulting in fixed action patterns is called innate releasing mechanism, IRM. The term innate releasing mechanism was coined by Nicolaas Tinbergen and is synonymous with Angeborene Oslo's mechanismus, ARM, coined by Konrad Lorenz. IRM sometimes also refers to the region of brain that gets activated in response to a particular sign stimulus. IRMs are of two types, i.e. closed and open. 3.7.1 Closed IRMs. These are pre-programmed fixed motor patterns that become functional as soon a sign stimulus initiates neural circuitry and they are unchangeable in response to environment. Closed IRMs include many courtship signals and its responses. Example, male rufous grasshopper Gompha cerupus rufus, Fig. 3.12, performs a variety of courtship behaviors that include head rocking, antennal flicking, stridulation and loud song. This entire routine is performed again and again in the same pattern in front of the female, either till she accepts his proposition and copulates or until the male tires down completely and goes elsewhere. If not accepted by her. This behavior during courtship is performed like a song, playing on a loop. 3.7.2 Open IRMs. In many species, the instinctive behavior that is being performed for the first time when exposed to stimulus can be modified or improved with multiple exposures and interactions with the environment. Such IRMs are called open IRMs. Example, in 1969, Haleman did multiple experiments on the laughing gull chicks. In one such experiment, he exposed them to cardboard model of the laughing gull and herring gull. Initially chicks pecked all models, but after few exposures, experienced chicks only pecked model closest to laughing gull head and ignored others. This improvement in behavior helps the chick to modify the behavior for its own advantage. SAQ3. A. Define key stimulus. B. What is innate releasing mechanism? C. Differentiate between closed and open IRM. 3.8 Stimulus filtering. This is the mechanism in which an organism, when exposed to multiple stimuli, simultaneously is able to elicit response to sign stimulus at the cost of another stimulus which are ignored. It is a phenomenon when brain fails to respond to a stimulus which under normal condition elicits a response. This happens when nervous system is able to distinguish the minor differences between the stimuli input and is able to prioritize response to sign stimulus over others. We will discuss few examples of stimulus filtering in this section. Midshipman fish mating behavior. Midshipman fish has auditory receptors that can detect sound waves between 60 to 120 Hz, which are dominant sound signals underwater. During breeding season, male courtship involves humming song in auditory range of 400 Hz. The breeding season also causes changes in female physiology and auditory receptors, making them receptive towards higher frequency male humming. At this time, rest of the sound signals are ignored in favor of male humming song. Key stimulus that translates into species reproductive success. Detection of bats ultrasound by moths. Moths and bats are nocturnal creatures and have prey-predator relationship. Bats feed on moths and moths use multiple tactics to avoid bats' echolocation. Moths have 2 ears on either side of their thorax that are capable of receiving ultrasonic signals from bats. Fig. 3.13. Look at the screen. Fig. 3.13. Placement of ear, auditory receptor in a moth. These ears are equipped with two auditory receptors A1 and A2 that are attached to tympanum in the ear. Fig. 3.14. When moth comes in contact with bat's ultrasonic waves, they sweep over moth's body causing vibration of tympanum and modification of A1 and A2 receptor cells. Stretch sensitive. Channels open up in the plasma membrane of these receptor cells, changing the permeability of the cell membrane and firing of receptors. The A1 receptor is the main receptor in moths and it gets fired at low frequencies of sonar. Fig. 3.14. A2 is the backup receptor that gets fired on receiving high-frequency sonar and works during emergency. Fig. 3.14. Situation A. Bat is very far away and moth receives signal from bat's echolocation. At this time, since signals are coming from far away, they will be low in frequency and will activate only A1 and moth responds by flying away from that place. Fig. 3.14. Situation B. Bat is very close to the moth and the moth receives very high frequency signal from it. A2 receptor fires up and moth initiates erratic flight movements to evade bat. The different sonic sensitivities of moth's A1 and A2 receptors helps it to adjust to bat vocalization via stimulus filtering. For long-distance evasion tactics, signal from A1 is utilized and short-distance evasion tactics uses A2 activation. Fig. 3.14. This physiological mechanism aid moths escaping predation by bats. Fig. 3.14. Response of A1 and A2 auditory receptors in moth's ear. Look at the screen. 3.9 Supernormal stimuli. The above normal level of sign stimulus is called supernormal stimulus. A stimulus under regular circumstance will elicit a normal response or behavior in recipient organism, but under artificial condition or during stimulus manipulation, organisms are exposed to exaggerated stimulus that will generate a very strong behavioral response in them. Examples: the gape color of chicks indicates the health of the chicks. Immune system. This is also a sign stimulus for parents and instigates them to feed the chicks. When in barn swallow, chick's gape color is reddened using food colors. The chick with red color gape were overfed as compared to yellow chicks. In this case, artificial red gape color works as supernormal stimulus. In nature, it is used by brood parasite chicks to get fed by the small-sized foster parents. The oversized chick of common cuckoo mimics begging signal of reed warbler host parents. 3.10 Fixed action pattern. Fixed action pattern are the simplest kind of instincts exhibited by various animals. These behaviors are hardwired into an organism's nervous system and are mostly stereotypic in nature. FAP always follows a well-orchestrated sequence of events once an organism is exposed to the key stimulus. These patterns are produced by innate releasing mechanism and are highly species specific. 3.10.1 Fixed action pattern characteristics. Konrad Lorenz founded the concept of fixed action pattern, FAP, and recognized its 6 key characteristics. 1. Stereotyped behavior follows the same predictable steps at every exposure to stimulus. 2. Complex. There are multiple interwoven complex steps in this behavior. 3. Species-specific characteristics. All members of same species demonstrate similar behavior. 4. Released. Certain sign stimulus is able to generate FAP and similar set of stimuli will trigger same response. 5. Triggered. Once the behavioral response starts, it won't change even though there may be changes in key stimuli or other factors. 6. Independent of experience, it is not learnt. Egg rolling behavior in female grey-lag goose. In 1939, Lorenz along with Tinbergen studied egg rolling behavior in grey-lag goose, where they were able to present all characteristics of FAP in the above-mentioned behavior. Grey-lag goose is a ground-nesting bird that makes its nest among reeds and shrubs. Grey-lag goose lays 4 to 6 eggs per nest and the female incubates the eggs till they hatch. It was observed that if from the clutch, an egg rolled outside of the nest, the female using her beak will roll and bring it back inside the nest. This displacement of egg works as key stimulus that initiates fixed action pattern leading to rolling of egg till it gets inside nest. Fig. 3.16. Look at the screen. Fig. 3.16. Egg retrieval behavior in grey-lag goose. Experiment conducted by Lorenz. Although physically the egg was not there, mother goose still does all the necessary beak actions that is required to roll it back as if it was present there. Experiment No2. Mother goose was introduced to egg-shaped objects such as doorknob or golf ball and it will roll it back to its nest. A model of large egg was used to initiate FAP in grey-lag goose. Although it is too big to be laid by her, she still tries to bring it back to nest by rolling with her beak. Inference. This manipulation of sign signal by scientist proves the ballistic nature of this response. It illustrates that once triggered, FAPs will be completed irrespective of the changes in the environment. The neuronal network that regulates this behavior in goose was given the name innate releasing mechanism, IRM, by Lorenz and Tinbergen. The model of large egg in this experiment works as a supernormal stimulus that triggered stronger response in the female goose. Aggressive behavior of the male of 3-spinned stickleback fish. Another model system for fixed action pattern used by Tinbergen was 3-spined stickleback fish. Breeding season brings physiological changes in the males of 3-spinned stickleback fish that include their bellies turning red and display of very aggressive behavior towards other males. Males during courtship build nests to attract females in their territories and defend both nest and territory from other male stickleback fishes. Tinbergen used cardboard model of fish and exposed it to male stickleback fish. Fig. 3.17. The model included all shapes of fishes with either red-bellied or swollen-bellied. The male fish attacked all red-bellied model irrespective of shape and tried to mate with the swollen-bellied one. The ferocity of attack on models increases with the increase in the red color of the belly. Look at the screen. Fig. 3.17. Male aggression towards cardboard models. This experiment proved that the red belly color is working as sign stimulus that Activated innate releasing mechanism in male fish leading to attack on the red-bellied model, Fig. 3.18. Fig. 3.18, male stickleback fish showing aggressive behavior toward red-colored belly. 3.10.2 Fixed action pattern, evolutionary significance. Evolutionary advantages. The fixed action patterns are the simplest but most effective instincts. They provide advantage in the form of fast response to stimulus and completion of task. This behavior is very important for the survival and fitness of an organism or its young ones. In case of grey-lag goose's egg retrieval behavior, survival of eggs depend upon quick response and making effort every time the egg rolls out of the nest. The lack of modification and sheer singleness of the purpose keep mother goose behavior consistent for every generation of offspring and help in their survival. Similarly, male stickleback's behavior helps him to defend his territory and nest and improves its reproductive success by improving the mating chances. Most fixed action patterns are generated in response to stimulus that require swift response and are important for survival or reproductive fitness. Evolutionary disadvantages. Some species learn to take benefit of other species' fixed action patterns by mimicking their sign stimulus. This process is called code breaking. Fixed action patterns are species-specific responses, and duplication of their sign stimulus make them susceptible, leading to loss of survival or reproductive fitness. Most famous example of code breaking is brood parasitism, where one species tricks another species to provide parental care to its offspring. 3.11 Code breaking. The mechanism of duplication of one species' sign stimuli by another to take advantage of the first species is called code breaking. Examples include brood parasitism and mimicry. 3.12 Brood parasitism. Brood parasitism is seen mostly in birds. Birds' chicks require lots of care for survival, which is a daunting task for the parents. Few species skip this laborious work by tricking other species to take care of their offspring. Chicks of parasitic species mimic the sign stimulus of host species and mislead them into thinking of the parasitic chick as their own. Parasitic species usually provide supernormal stimulus to foster parents. This forces them to provide extensive care to the foster chick. Examples. The common examples include common cuckoo parasitizing variety of birds such as dunnock, common crow and reed warblers. Cowbirds are also brood parasites that are known to parasitize sparrows, eastern phoebe and masked water tyrant. The cowbird leaves its egg in the nest of the host bird, which is larger in size compared to the host. The cowbird chick hatches first, is larger in size and has the same food-begging behavior as the host chicks. Fig. 3.19. This works as supernormal stimuli for foster parents. Rufous collared sparrow, and they feed this oversized chick till it leaves the nest. Look at the screen. Fig. 3.19, shiny cowbird chick, left, fed by rufous collared sparrow, right. 3.13 Mimicry. This is an evolutionary adaptation in which one organism bears a resemblance to either another organism or inanimate objects. The organism whose physiognomies are being duplicated is called model, and the one that is trying to resemble the model is called mimic. The characteristics that are duplicated include physical appearances, acoustic signals, tactual stimulus and chemical signals. The mimic sometimes copy more than one feature of the model organism. The most common objective of mimicry is protection from predator, but it can also be used for other purposes like reproductive advantage. Predation and parasitism by mimic species. Many animals mimic non-living objects like stones and fallen twigs, tree bark, leaf and even flowers. In these cases, model are often called as mimesis or masquerade. Example, orchid mantis, Fig. 3.20, looking like an orchid flower, and mimic the ability of absorption of UV rays like actual flower. For the pollinator, the flowers in UV light, seen as blue-colored, works as sign stimulus, and they get attracted toward them. The orchid mantis, using code breaking, copies same stimulus and preys on the unsuspicious pollinators. Plant hoppers, these insects, Fig. 3.21, are like grasshoppers, but they look like leaves of the plants and camouflage among them. 3.1 Mimetic releaser. Animals are sensitive to sign stimulus that initiate fixed action pattern in them. This relationship between the sign stimulus and reaction exhibited by animals is exploited by variety of species for its own benefit. The mimic stimulus or signal of exploiter species is called mimetic releaser. Example, many fishes are dependent on cleaner fish for removal of ectoparasite, dead skin and fungi from its body. The true cleaner fish uses undulating dance as a signal of arrival that causes fixed action patterns in client fish to align in a cleaning position. Examples include blue streak cleaner, wrasse, Labroides dimidiatus, who works as cleaner fish, Fig. 3.22a, for surgeon fish, moray eel and Acanthurus mata. This behavior of cleaner fish is mimicked, aggressive mimic, by blue-striped fang blenny, Plagiotremus. Rhinorhynchos, Fig. 3.22b, that poses as cleaner fish and instead takes away skin and scales of the unsuspecting client fish. The stripe and color pattern on the body of blue-striped fang blenny, Fig. 3.22b, work as mimetic releaser, initiating FAP in client fish causing their alignment in cleaning position. Fig. 3.22a. Blue streak cleaner wrasse cleaning client fish, moray eel. Fig. 3.22b. Blue striped fang blenny mimicking blue streak cleaner wrasse. 3.13.2 Classification of mimicry. Defensive mimicry, aggressive mimicry, reproductive mimicry, auto-mimicry, defensive mimicry. In this type of mimicry, an organism avoids harmful animals, usually predator. By mimicking the model, animals safeguard themselves from predators with this type of mimicry. Classification of defensive mimicry. Batesian mimicry. This mimicry was described by English naturalist Henry Walter Bates and hence the name Batesian mimicry. In this type of mimicry, a harmless species usually imitates a harmful one and avoids predators, anti-predator adaptation, in the process. The model species usually remain unaffected. Many species have developed anti-predator adaptations like presence of toxins in wasp, venom in snakes and skunks using obnoxious odor or bitterness in many insect larvae. All the predator averting mechanisms are signaled by bright coloration and its intensity indicate level of deadliness. Mimic species do not invest in toxin, but instead imitate aposematic patterns of harmful animals. This usually deceives the predator that avoids both model aposematic species and its mimic. Examples: visual mimicry, common Mormon butterfly, Papiliopolites, Fig. 3.23a. Mimics the unpalatable common rose butterfly, Fig. 3.23b. In a habitat, the population of common rose model is much higher compared to population of common Mormon. This gives predator enough encounters with the unpalatable common rose and later it avoids both common rose and common Mormon equally. This way mimicry gives protection to harmless palatable common Mormon butterfly from predators. Look at the screen. Fig. 3.23a. Common Mormon butterfly, Papiliopolites. Fig. 3.23b. Common rose butterfly, Pachliopta aristolochii. Fig. A common Mormon butterfly, top, mimics common rose butterfly. Bottom. Acoustic mimicry. Bats are nocturnal animals and known to find their prey using echolocation. Not all moths are edible and the inedible moth like tiger moth Cesnea tenor, Fig. 3.24a, sends ultrasonic warning signals to big brown bat and red bat. Bats over the period of time learn to avoid inedible tiger moth. Edible parallid moth, Fig. 3.24b, mimics tiger moth, Fig. 3.24a, by producing similar ultrasonic signals and is also shunned by the bats. Fig. 3.24a. Tiger moth, model, Fig. 3.24b. Parallid moth, mimic. Electrical mimicry. The electric eel kills its prey by giving it a strong electric shock. Electrolocation discharge created by electric eel gets mimicked by blunt-nosed knife fish, Brachyhypopomus. Blunt-nosed knife fish produces similar electric discharge patterns as electric eels and remain protected from their predators. Mullerian mimicry. This mimicry was described by German naturalist Fritz Müller and named as Mullerian mimicry. In this type of mimicry, two unrelated Harmful or unpalatable species mimic each other's aposematic patterns or signals to warn their common predators. In this case, both species possess honest aposematic signals like toxins, venom, or auditory signal and mutually benefit from each other's mimicry behavior. This is also a good example of mutualism, and both species invest in toxin or defensive chemicals. A naive predator initially kills small number of unpalatable prey before learning not to attack them. This process causes death of only a few members of unpalatable species population. With two unpalatable species mimicking each other, they share the load of predator education, and thus death of unprofitable prey is divided between two species, rather being borne by only one. Examples of mullerian mimicry: viceroy butterfly, Fig. 3.25, are foul-tasting to its predator, and its mimic monarch butterfly, Fig. 3.25, is equally unpalatable. Since they share common predator, it helps both the species to reduce predator-related deaths. Relationship with Batesian mimicry. Mullerian mimicry and Batesian mimicry are contrasting types of mimicries. In Batesian, harmless species avoids predator at the cost of Batesian model and represents parasitic relationship between model and mimic. On the other hand, as both model and mimic get benefited, mullerian mimicry is example of mutualism. In nature, you get co-mimics that utilize both Batesian and mullerian mimicry. Wasps and bees mimic each other and they both can sting, whereas hoverfly just mimics wasps and utilizes their stinging capacity for its protection. Fig. 3.26. Fig. 3.26. Relationship between Batesian and mullerian mimicry. Emslian mimicry, Mertensian mimicry. This was first suggested by M.G. Emslian and later expanded by biologist Wolfgang Wickler, who named it after Robert Mertens, German herpetologist. This is known as both Emslian and Mertensian mimicry. This is a unique type of mimicry, where more harmful or dangerous species mimics the less harmful species. Example: this is seen in case of deadly coral snake, Fig. 3.27a, mimicking bright color pattern of moderately toxic false coral snake, for example, Erythrolampras aesculapi, Fig. 3.27c, that is also model for harmless Mexican milk snake, Fig. 3.27b, Batesian mimicry. In this case, the more dangerous species is lethal to the predator, and if it encounters the mimic first, it will die along with prey. Predator death leaves no scope for learning and avoiding the prey. The highly poisonous snakes mimic less harmful one, and the predator usually learns its lesson by encountering the model. Consequently, it will avoid all the bright-colored snakes in the future, safeguarding the lethal mimic. Wasmannian mimicry. Austrian entomologist Erich Wasmann proposed Wasmannian mimicry. In Wasmannian mimicry, model animal and mimic share the colony or nest where they live and are in symbiotic relationship, commensalism, with each other. Ant mimicry is very good example of it. Everywhere in world you will find abundant population of ants, and due to their aggressiveness and unpalatability, predators avoid them. Spiders that live commensally in ants' nest mimic them and remain protected from the predator. This is also a Batesian mimicry. Vavilovian mimicry. This is observed in plants and also known as crop mimicry or weed mimicry. Named after Russian scientist Nikolai Vavilov, this mimicry proposes that the weed mimics morphological features of domesticated plants for its own reproductive success. In this case, human gets deceived into taking care of weeds that looks like a domesticated plant and grows along with it. The mimic gets the advantage of mutualistic relationship between human and artificially selected plants and showcase indirect artificial selection. Example: the example of Vavilovian mimicry is rye and oats, plants that are mimic of wheat and barley. Now a separate and important crop. Initially they were weeds of wheat and barley. Gilbertian mimicry. This is the kind of mimicry where you observe reverse host-parasite aggression. The host mimics the parasite or the prey mimics the predator in order to avoid getting exploited by them. This kind of mimicry develops after multiple adaptations and strong selection pressures. It is seen in case of passion flower that contains toxins to defend itself from herbivores. Example, the heliconius butterfly larvae developed enzymes that can break down passion flower toxins and they selectively parasitized it. To save themselves, plants developed stipules that mimic the eggs of heliconius butterfly. The larvae of heliconius butterfly are cannibalistic in nature and they older larvae eat younger ones. Presence of stipules discourages butterfly to lay eggs close to it and thus these egg mimics protect the plant. Often egg mimics are also nectarines and they attract predators of butterfly larvae. Browerian mimicry, Lincoln P. Brower and Jane van Zandt Brouwer proposed this mimicry and it is a kind of auto-mimicry. In this less toxic, non-toxic members of species mimic toxic members of the same species. Example, the larvae of queen and monarch butterfly, Fig. 3.30, feed on variety of milkweed that has varied level of toxins. These butterfly larvae are able to store toxins in their cells and even transfer it to adults. The variation in the level of toxins in the diet of larvae bring variation of toxicity in larvae and adult form. Therefore, less toxic larvae mimic the more toxic members' patterns to protect them against predators. Aggressive mimicry. This type of mimicry is offensive mimicry exhibited by predators and parasites to duplicate sign signals or patterns of a harmless animal to deceive its prey or host. Sometimes predator exploits sign stimulus of a prey and by mimicking it the predator lures them into its trap. George and Elizabeth Peckham suggested an alternative term for this type of mimicry as Peckhamian mimicry. Classification of aggressive mimicry. Luring prey. This type of mimicry involves predator mimicking sign stimulus of prey and luring them towards itself. Example, the small wildcat mage mimics the voice of crying pied tamarin infant. This sound attracts adult pied tamarin towards mage. Once pied tamarin is within the range of cat, mage hunts it down. Appearance of food. The aggressive mimic in this type of mimicry camouflages itself as food of prey and hunt unsuspecting prey that comes for nourishment. Example, the tongue of alligator snapping turtle resembles a pink colored worm and it attracts many fishes towards it. As soon the fish approaches this camouflaged predator, it eats them. Spider Argiopa dentata weaves a zigzag pattern in its web that resembles nectar guides of flowers seen under UV light. This attracts bees in hope of nectar and they instead get trapped in the spider web. Bipolar mimicry system, the mimicry system that involves two specific species, where one species is the mimic and second one is the dupe. Its subtypes include Batesian volation mimicry and brood parasite mimicry. Batesian volation mimicry, named after Henry Walter Bates and Alfred Russel Wallace, it is also called prey mimicry. In this type, model is the female prey that is sexually receptive, mimic is the predator and dupe is the male prey that gets deceived by the mimic. Examples, female bolus spider produces sex pheromones of moths, normally produced by female moths, and lures the male moth using them. The spotted predatory katydid mimic sends sexual clicks same as that of the receptive female cicadas. These clicks attract males in search of females and they become target of katydid attack. Brood parasitism mimicry. Brood parasitism is where parasite species dupes the host and makes them raise their offspring at the cost of host's own offspring. This deceit involves parasite mimicking the morphology of hosts' eggs. Example, the cuckoo is a famous brood parasite that parasitizes multiple species. When cuckoo parasitizes reed warbler, the egg produced by cuckoo, although larger in size, is the mimic of reed warbler eggs in color and pattern. Also, the adult female cuckoo mimics, Fig. 3.36, the vocalization of predator. Sparrowhawk, Fig. 3.36. This scares the small birds like reed warbler away from nest and gives time to female cuckoo to lay egg in it. Wicklerian Eisneron, this is also called as mimicry of harmless species, as the predator mimics the harmless species to fool the prey. Mimic model in this mimicry also includes a mutualistic ally of the target species. Example, blue streak cleaner wrasse, cleaner fish, is used as model by mimic. Saber-toothed blenny, false cleaner fish. The blenny poses as cleaner fish and gets away with flesh and scales of unsuspecting client fish in The process. Mimesis. In this mimicry, the mimic imitates a modular species towards which prey is indifferent. In this case, predator closes on to the prey unnoticed and attacks it when opportunity arises. Example, the turkey vulture is used as model by zone-tailed hawk that hides among turkey vulture while flying and attacks unsuspecting prey when it's close enough. Parasite mimicking host prey. In this case, the roles get reversed. The parasite mimics the host's prey, and as host approaches the parasite in hope of food, the parasite enters its body and completes its life cycle inside it. Example, freshwater mussel, Lampsalis-08 uses its mantle to look like fish and lure largemouth bass, a predatory fish, towards itself. Once the predatory fish attacks, it ejects its larvae into its mouth. Larva latches onto the fish gill, takes blood meals to complete its cycle and leave fish as an adult. Reproductive mimicry, animals mimicking members of either other species, or in some cases same species, to accomplish successful mating, reproduction. Classification of reproductive mimicry. Bakering and Dodsonian. Bakering mimicry, it is named after British-American botanist Herbert G. Baker and is also an example of automimicry. In this, the female flower resembles the male flower of its own species, but unlike male flower is devoid of nectar. The pollinators get attracted towards it and pollinate the female flower in hope of nectar. But they get duped as they were conned out of the reward, nectar. Dodsonian mimicry, proposed by American botanist Callaway H. Dodson. This mimicry also involves pollinator and a non-rewarding mimic flower. But in this case, the model and the mimic belong to two different species. Flowers of epiphytic orchid, Epidendrum ibaguense mimic flowers of tropical milkweed Asclepias curasavica to get pollinated by humming birds and monarch butterfly. Pseudocopulation, many flowers mimic the females of the pollinator species to attract male insects. As the male tries to copulate with the mimic flower, it ends up pollinating it. Morris Alexander Poian described this mimicry, and it is also called as Poianian mimicry. Example includes mirror bee, orchid and fly orchids that mimic their pollinator bees and flies, respectively. Intersexual mimicry. In this type of mimicry, an individual of one sex mimics the opposite sex of same species to achieve mating. This happens where intrasexual competition is very high. Usually the male exhibits this mimicry, i.e. pretending to be female so that they get a chance to be close to an actual female. Example, common side-blotched lizard is very aggressive and males jealously defend their territories. There are three morphs of males in this species. 1. Orange-throated males. They have orange coloration in their throat. They are the most aggressive, ultra-dominant and largest size males among the male morphs. They keep harem of females for mating purpose. The yellow-throated male mimics who enter their harem, pretending to be females, fool them. 2. Blue-throated males. They have blue coloration in their throat and medium-sized males usually mate with single female. They are able to catch the yellow-throated mimics, but susceptible to losing mate to aggressive orange-throated. 3. Yellow-throated males. These males mimic the yellow coloration of throat as exhibited by female to reach to the guarded female. They also mimic the rejection sound produced by females to create a convincing deceit. They are perfect example of intersexual mimicry. Automimicry. This is intraspecies mimicry where organism mimics and exhibits another part of its own body. Examples, four-eye butterfly fish mimic its own eye spots and exhibit them on its tail as false eye spots. The mimicry deflects predator's attention from its vulnerable head towards tail and gives it precious time to escape. Similarly, pygmy owl spots false eye spots on the back of its tail to look aggressive and misguide the predators. 3.13.3 Evolutionary benefits of mimicry. It is considered to be a positive adaptation. The defensive mimicry and auto-mimicry helps prey to escape predators. But for the Batesian mimicry to work, the number of models should be far higher compared to the number of mimics in a particular habitat. The aggressive mimicry showcases the prey-predator relationship and deceit tactics used by predator to lure prey. The reproductive mimicry helps finding innovative ways to keep up to the sexual competition and find suitable mate. Thus, mimicry showcases the natural selection of resemblance and multiple ways it is being utilized by mimics. 3.14 Summary. In this unit, you have learned that innate behaviors are inbuilt behaviors that are hardwired in the nervous system of an organism. Examples of innate behavior include spatial orientation, reflexes, motivation, and instinct. Instincts are those behaviors which are performed by an animal on its first encounter with the stimulus. They do not require any prior experience. It includes mating behavior, parental behavior, escape from predator, etc. Any environmental signal sensed by animal is termed as stimulus, and stimulus that initiates fixed action patterns in an animal are called key stimulus. Key stimuli are able to produce fixed action pattern in animals by complex neuronal network known as innate releasing mechanism or IRM. Those innate releasing mechanisms, which remain unchanged, are called closed IRM, and those that get modified based on past experiences are named open IRM. All animals are constantly exposed to multiple stimuli. Animals do not act on all of them but rather choose to respond to only few important ones. This mechanism is called stimulus filtering. Supernormal stimulus is key stimulus of a species imitated and exhibited at very high level by some other species to reap benefit from the original species. Fixed action patterns are patterns produced by innate releasing mechanism in animals in response to key stimulus. Code breaking is the mechanism by which one species emulates sign stimuli of another to take advantage of first species. Its examples include brood parasitism and mimicry. In brood parasitism, parasite species deceive host and make them take care of their progeny. Mimicry is an evolutionary adaptation where one organism bears a resemblance to either another organism or inanimate objects. This is being used to escape predator, lure prey, and find mate for sexual reproduction. Thank you.